Hello friends, welcome to this class of biology where we are going to discuss different aspects of molecular basis of inheritance. Nowadays from the school level we are acquainted with the term genes, genetic engineering and biotechnology, Mendel's laws of inheritance and many more things. But at the present context we must know what genes are. If we look at the history of genetics, that will give us certain concept how the concept of gene and the search of genetic material, what are genes are made of what exactly, the history will give us many important aspects. So, now I am going to discuss the history starting from Mendel's period. Mendel, who is regarded as the father of genetics, he spoke of the factors. The factors, they are discrete particles. Mendel in 1865 he coined the term factor. So, these factors are discrete particles, but then only after towards 1880 chromosomes were discovered. Chromosomes. So, discovery of chromosomes towards 1880. Then towards 1903 homologous chromosomes were discovered. That means chromosomes exist as homologous pairs, and we might have heard that chromosome theory of inheritance. Mendel's theory of inheritance and chromosome theory of inheritance, they go in parallel. One of the important proponent of this chromosome theory of inheritance is Walter Sutton. During this time of 1903, what he proposed, that as factors inherit, similarly the chromosomes also. Therefore, he went on emphasizing that genes are present in the chromosomes, but thereafter discovery of crossing over and linkage by Thomas and Morgan in Drosophila gave in-depth knowledge regarding these genes. We know a linkage group, a chromosome consists of, or it has many genes arranged in linear fashion. Then only a chromosome consists of two important kind of macromolecules, one is DNA and the other one is histone proteins. DNA and histone proteins, they comprise the eukaryotic chromosome. Towards 1920, it was found that proteins are composed of around 20 amino acids. Protein diversity and complexity is very high in comparison to DNA. The DNA consists of only 4 kinds of nucleotides. 4 nucleotides comprise or make the DNA, but proteins are made of 20 different types of amino acids. So, it is Linus Pauling, one of the great biologist, chemical biologist, who proposed that protein might be, because of their complexity, diversity, be the genetic material. It was his proposal, but he could not arrive at any conclusion. But in the meantime, Frederick Griffith towards 1928, he went on experimenting on Streptococcus, one of the pneumonia causing bacterium. The Streptococcus earlier was called Diplococcus pneumoniae, but subsequently the name was changed to Streptococcus pneumoniae. This bacterium exists in 3 immunological forms depending on the presence of a carbohydrate in their cell wall. Each of the type 1, type 2 and type 3 type of pneumococcus, they were further categorized as S form and R form. S stands for smooth and R stands for rough. Why it is called smooth? Smooth form of bacteria, they possess capsule around them, and that capsule enables that bacteria to escape phagocytosis of the host, either it is a mouse or the human being. As they escape from the phagocytic effect of the human monocytes or mouse monocytes, they cause disease and they are called as virulent. And these S form of bacteria, they form smooth edge colony. That means, when they are plated in the Petri plate, they form smooth edge, but those without the capsule, they form rough edge colony, that is why they are R type. Therefore, all the types type 1, type 2, type 3 immunological types, they have both S and R form. One thing one must be remembering here, that every type, that means type 3 has S form as well as R form, and they are interconvertible. Next. Now, what Griffith did, he went on experimenting on this. He first injected type 3S pneumococcus to the mouse, and the mouse succumbed. It was subjected to pneumonia and died. But another form, it was type 2R. When it was given type 2R live, when it was given, the mouse also survived. In the first type 3S, mouse died, type 2R, mouse survived, and in the third one, what he did, he heat killed the type 3S bacterium pneumococcus and then injected to the mouse, and the mouse survived. That means, heat killed 3S pneumococcus upon injection to the mouse was not affected, it survived. But the combination of type 2R, in which the mouse survived, type 3S heat killed, in which also, that is in the diagram C, the mouse also survived. When both the combinations type 3 heat killed and live 2R were injected to the mouse simultaneously, then after some time some days it was found the mouse succumbed, it died, and from the dead body of the mouse, both live 2R and live 3S were recovered. It was a astonishing type of result. He injected dead 3S and live 2R, but from the dead bodies, both live 2R and live 3S were recovered. And what might be the result? He repeated this, then he put the questions, is it the recovery of the live 3S from the dead bodies, is it because of during heating some survived heating or some get back their life? It is not possible. Second one, it is not possible that any type 2R also mutated 3S. Griffith tried to justify or analyze what might be the region of appearance of type 3S bacterium from the dead body of the mouse, because it did not inject live 3S, but how they came. So, that is why the first proposal that all were heat killed and none survived, that was okay, and the second analysis, is there any possibility that 2R may mutate to change to 3S? It is not possible, because in each category mutation possible, type 2R cannot mutate to type 3S. Then his conclusion was, there is something, it passed from dead 3S to live 2R, changing the latter, that means changing the live 2R to 3S, and he could not conceptualize what that something. So, he conceptualized that there must be some transforming principle, there must be some chemical which transform 2R cells to some of the 2R cells to 3S, so that they appear therein. Okay. But there it ended, because it was a surprising discovery. With intention he did not go for, but he found there is a transforming principle which transform 2R to 3S. Then his experiment was carried out by another group of scientists, and they are Avery-MacLeod and McCarty. They furthered the experiment of this person Griffith. They repeated this and they found the same result. What Griffith did? They got similar results after repetitions. Then they thought of doing experiment in vitro. What they did? They took type 2R cells, and their experimental setup was that they killed the 3S bacterium, extracted the DNA from that 3S bacterium. In the test tube they put 2R bacterium, 3S DNA, and then enzyme treatments were done. If you can see this diagram, when 2R is cultured, it gives rise to 2R colonies. Only the extract of DNA extract of 3S, when it was put in the petri dish, no colony. In the third category, when in permutation it was given 2R plus 3S extract, DNA extract from 3S, and enzyme combinations were given. Why enzymes? Because during that time the purification of DNA was a difficult process. They thought there might be impurities with DNA of proteins or RNA or any other chemical. That is why what they did, they treated these combinations with different enzymes. When no enzyme is given, 3S colonies appeared. When RNase is given, RNase means RNA degrading enzyme. If any RNA in DNA extract is present, that would be degraded. That means, in presence of treatment of RNase, 3S colonies appeared. When DNA was given, DNA digesting enzyme, DNA hydrolyzing enzyme was given, then no colonies appeared. Trypsin, which is a protease, when it was given, 3S colonies also appeared. It was DNA which blocked the process of transformation. This proved that DNA is the transforming principle. So, now, transformation of the non-virulent 2R bacteria was blocked only if DNA was destroyed, suggesting that DNA was carrying the information required to transform the 2R to 3S. The publication of these studies by Avery-MacLeod and McCarty that appeared in 1944, that provided the first evidence that DNA is the genetic material, but thing is they did not emphasize well, or they did not report to the scientific community it is DNA rather than protein as genetic material, but their experiment was very definitive experiment, but claim was very poor. Now, following that as their experiment was good, but claim was very low, or it was not emphatic, but after 7 years towards 1950-51, viral geneticist Hershey and Chase, they experimented on T2 bacteriophage. T2 bacteriophage, which is the bacteriophage that parasitizes the host cell E. coli, and at that time the bacteriophage are well visible under electron microscope, and electron microscope studies revealed that certain things or a big structure is remaining outside the bacterial cell and certain things are going inside. But this T2 bacteriophage is having a genetic material packaged in its head, and it is surrounded by a protein coat. And what they did, Their objective was to differentially label the proteins and DNA. They know, at that time it was known that proteins have a unique chemical or unique element, that is sulfur, and they labeled the protein coat with AS35, and in another set they labeled the DNA with the unique element, that is phosphorus. That means, they labeled two batches of viruses, one with AS35, one with P32, as a result of which they did this. So, now the very meticulous experiment of MacLeod, McCarty and Avery over a period of 13 years resulted in a very clear understanding or clear finding that the transforming principle is DNA. But these three, they did not claim before the scientific community emphatically that DNA, they did not emphatically claim that DNA is the genetic material rather than protein. Their claim was very at very low level. Therefore, even though they experimented thoroughly that DNA is genetic material, because of their claim it was not surfaced before the scientific community that DNA is the genetic material, and but they proved that the transforming principle is DNA. Thereafter, the small phase, bacteriophage, T2 phase, type 2 phase was taken into experiment. Hershey and Chase, two viral geneticists, they worked on this, and by that time they know this bacteriophage consists of two kinds of molecules, one is DNA, another is protein. The protein is the outer structure, or protein encloses the DNA in its head, and the structure of virus T2 was well studied because of the availability of electron microscopy, and this T2 phase parasitizes its host E. coli, and it replicates within that and produces progeny phases within a very short span of time. This was known to these scientists, and with this they experimented further to unravel or to know that DNA as the genetic material. As electron microscopy became a very good tool for study of bacterial genetics and viral genetics, Hershey and Chase, they studied that this bacteriophage consists of two kinds of macromolecules, one is your protein coat and the other one is the genetic material, and the genetic material is housed inside the protein coat, and what is seen that this the genetic material is present in this. It is a condensed structure, the DNA is condensed within this head. Now, this protein is, this is protein, and when the chemical composition of protein was studied, it was found that protein contains a unique element, that is sulfur. So, sulfur is unique to protein, and this DNA, the nucleic acid contains, that is DNA contains a unique element, that is your phosphorus. So, their logic was to label, to make label the two kinds of macromolecules. So, protein was labeled with AS35, that is radioactive sulfur, and phosphorus was made radioactive by P32. So, DNA was labeled by P32 and protein was labeled by AS35. So, what they did in two cultures or in two batches, they made bacteriophages labeled, one with AS35, that means protein was labeled in one set, the other set the DNA was labeled with P32. So, how they did, how virus can be labeled? Actually, in one flask, conical flask, they supplemented with phosphorus P32 and grew bacteria, and thereupon they cultivated with bacteriophages. So, these became labeled with P32, but in the other one they were labeled with sulfur, and in their course of experiment what they did, they allowed these bacteria, these phages to infect new bacteria, new E. coli cells, and these infected bacterial cells were subjected to shearing force, centrifugal force by whirring blender. And from the previous classes or previous knowledge, we know bacteria is, or a phage infects a bacterial cell by sitting on its. So, it adsorbs to the surface of bacterial cell. When the bacterial cells infected with the viruses, they are subjected to shearing force of a whirring blender, then the phages are shed off from the bacterial cells. So, the phages are separated from the bacteria after certain minutes of time, then they were subjected to centrifuge, and as a result of which in the centrifuge tube two portions were there, heavy bacterial cells, they were settled to the bottom, they formed the pellet, and in the supernatant the viral coats remained. So, now look at this diagram. In this diagram there are two batches. In one batch, in the diagram A, that is labeled with P32, and ultimately at the end the P32 appeared inside the bacterial cells, because the bacteria they settled to the bottom upon centrifugation, and the radioactivity was found in the pellet, and the bacteria on further production of new phages, and P32 appeared in the progeny phages. And in the other set, when S35 was labeled, allowed, these phages were allowed to grow on the bacteria, subjected to shearing force, centrifuged. The pellet did not have any radioactivity, rather radioactivity was found in the supernatant, the upper portion, and the upper portion is belonging to the coats, the protein portion. So, now in the second one radioactivity is found in the protein portion. In the first one radioactivity is was found on the pellet portion, the cell bacterial cell portion. So, therefore, they concluded that it is the genetic material after centrifugation. So, radioactivity in the supernatant and the bacterial pellet was determined, that I have already told you. When T2 phase containing P32 was mixed with host bacterium, most radioactivity was found in the pellet, that we went on discussing. Thus, radioactive DNA was injected into the cell and phages were produced, proving that DNA was carrying the virus's genetic material information. In the B, when E. coli was infected with T2 phase containing S35, most of the radioactivity remained in the supernatant, because the protein coat becoming light, they were sheared off by centrifugal force and remain in the supernatant. Nothing protein entered the bacterial cell. Therefore, they found most of the radioactive protein was released into the supernatant, whereas P32 DNA remained within the bacterial cell. Since genetic material was injected and T2 progeny were produced, DNA must have been carrying the genetic information for T2. Now, by this definitive experiment, a very scientific experiment, because they are rationale or the basis of experimentation was, they knew that phages reproduce within the bacterial cell and certain portions remain outside, and certain portions are injected into the cell. But what was injected into the cell and what remained outside the cell, that was distinguished by using P32 DNA and S35 protein, and therefore, they confirmed that DNA is the genetic material of the phages. What Avery and his group could not put forth before the scientific community emphatically, that is DNA as genetic material, but the final blow was given to the thought whether protein or DNA as genetic material. The experiment of Hershey and Chase proved that DNA as genetic material. So, now Griffith's experiment, let me tell you, Griffith experiment was a serendipitous or surprising discovery. He did not aim at getting such type of results, but he found that there is a transforming principle. Something is transformed, a chemical which enters from dead 3S cells to live 2R cells. So, that 2R cells are transformed, chemically modified, genetically modified, as a result of which this transformation occurs. And now, transformation discovered by Griffith is a very good step in transforming cells in genetic engineering. It is often used in injecting foreign DNA into bacterial cells and so also the eukaryotic cells. And the transforming principle was characterized chemically by Avery and his group. It was found to be DNA, but his low voice before the scientific community could not say that DNA is genetic material, but his experiment was wonderful, their experiment was wonderful. However, the differential radio labeling experiment, because differential radio labeling, why? Because phages in one group were radio labeled by S35, in other group they were radio labeled by P32. That proved DNA as genetic material. Now, let me tell you, up to this we came to know that DNA is genetic material. Whether RNA is any genetic material? Yes, we know RNA is genetic material in many cases. That is your first TMV, tobacco mosaic virus, that has RNA as genetic material. Retroviruses have RNA as genetic material. This coronavirus has RNA as genetic material, but it is the reconstitution experiment carried out subsequently that proved it is not the protein coat of the RNA, sorry, TMV, rather the helical RNA is responsible for producing its morphology. That means, reconstitution experiment in TMV proved RNA as genetic material, not protein. So, these 4 sets of experiment of Griffith, of Avery, of Martha and Chase, and Franklin and Conrad prove that nucleic acids are the genetic material. Okay. All the cellular life forms in this world have DNA as genetic material. Many viruses have DNA as genetic material, only certain RNA have genetic material. I hope you people have understood this the topic search for DNA as genetic material well. This class we are going to study regarding the structure of a DNA. How the structure of a DNA double helix was discovered. This is a long journey. We know Frederick Mescher in 1869 isolated a substance called nuclein. For that he took salmon sperm nuclei, because these salmon sperm they have very big nucleus as the macrophages have. As they have very big nucleus, it is very good material for isolation of the nuclear substance, and he found that it is much rich in phosphorus, about 10% of its dry weight is phosphorus, and he named this substance as nuclein, but almost after 10 years Altman gave the name nucleic acid. But DNA in its present form, what we know, it took long years to be established. Almost after 50 years or so, Russian biochemist P. A. Levin, what he did, he studied that the DNA consists of 4 nucleotides. Two important findings of Levin found that DNA consists of 4 nucleotides, right. And another thing is that we say that is deoxyribonucleic acid. So, he also discovered that deoxyribose sugar is present in this DNA. Two important findings of this Russian biochemist who came in 90s to USA and worked at Rockefeller University Institute and discovered that DNA consists of 4 nucleotides and the type of sugar is deoxyribose sugar. But side by side we know that Griffith also worked on mice and Avery, then Hershey and Chase, they established DNA as genetic material. But what is the shape of DNA? What is its physical structure? But side by side its molecular structures are revealed. Let us look at the slide, and we know in the first year course the DNA consists of, or any nucleotide consists of 3 kinds of substances, 3 kinds of small molecules. One is phosphoric acid. This phosphoric acid H3PO4 at physiological pH, that means 7 and 7.4 of pH, this phosphoric acid ionizes to form phosphate. And the type of sugar, ribose, a pentose sugar present in RNA and its ribonucleotides, deoxyribose sugar which lacks an oxygen atom in the second position, that is why it is deoxyribose sugar, and therefore, this is present in deoxyribonucleotides and the DNA as well. But here we are going to discuss on the 3-dimensional structure of a DNA, but prior to that, this of what the nucleotides are made of and how they are joined, we must have little bit discussion on this. So, now any nucleotide consists of base, sugar and phosphate. We discuss phosphoric acid and the sugar, that is pentose sugar, and 2 kinds of bases, they are purines and pyrimidines. Purines are double ring structures, pyrimidines are single ring structures. They are nitrogenous bases, or simply they are called as bases, because the nitrogen present in the purine ring or pyrimidine ring, because of their presence of their lone pair of electrons, behave like bases. That is why these nitrogenous bases are called as bases. So, purines are double ring structures and they are adenine and guanine. Pyrimidines are single ring structures, they are cytosine, thymine and uracil. DNA has adenine, guanine, thymine and cytosine, whereas in case of RNA, thymine is replaced by uracil, this we know. Now, what would be our discussion? A base, nitrogenous base is attached to a sugar by glycosidic bond, and the sugar is attached to, or sugar with its 5' carbon linked to a phosphoric acid forming a nucleotide. Here we can say that many nucleotides are joined to form a polynucleotide. Look at this polynucleotide, and there are 1, 2, 3, 4, 5, 1, 2, 3, 4. There are 4 nucleotides, 4 nucleotides. In each nucleotide, base, for example, cytosine is attached to the sugar in its first position, but in 5th position of the sugar it is attached to a phosphoric acid. So, this gives a nucleotide. This nucleotide is linked to another nucleotide, by means of covalent bond, and as that covalent bond is formed across phosphorus, 2 ester bonds alongside. That is why it is often told that nucleotides in a polynucleotide are linked by phosphodiester linkages, and when there is a chain, when there is a polynucleotide chain, you see this, because this diagram is very important in understanding. At one end there is 5 dash phosphorus, but the other end there is 3 dash OH. So, therefore, one terminus is having 5 dash P, the other terminus is 3 dash OH. Therefore, this polynucleotide is a polar structure, polar chain, 2 termini are different, and always the arrowhead is at 3 dash OH end, because it is the growing end. We will see that during DNA replication new nucleotides are added at this 3 dash OH end. So, now after having some knowledge on a polynucleotide chain and its constituents, what are the steps in, or what are the advancements in or towards achieving the discovery of the DNA double helix. So, in that direction, the first act was that of this lady scientist, this Rosalind Franklin. She was working with Maurice Wilkins, her mentor, in King's College London. They extracted DNA in a very pure form, bombarded X-ray to that DNA crystals, and found a particular pattern, and that particular pattern is called X-ray crystallograph, and this X-ray crystallograph was first of its kind in the history of science. And from this X-ray crystallograph, it indicates many things we cannot know. Those who are specializing on X-ray crystallography and its principles, they can say what this cross mark indicates. So, now Rosalind Franklin and Maurice Wilkins, they were slow in analyzing what this crystallograph speaks, but Watson, who was well trained in X-ray crystallography, he analyzed that it is a symmetrical structure. So, as it is the crystallograph of DNA, it is a helical structure with a uniform diameter, and it is around 2 nanometer, and there is a repeating units, or a helix repeats at a particular interval. So, this X-ray crystallograph gave many a clue to Watson and Crick for unraveling the structure of DNA. In addition to the X-ray crystallograph, then the two scientists, they studied also these two scientists, I want to say Watson and Crick. Watson belongs to America and he is English, Crick is an English person, but they did not go for any experiment, rather they studied the crystallograph of Maurice Wilkins and Rosalind Franklin, and subsequently they found very useful data of Avery-Chargaff, who studied the base composition of DNA, and studying different DNAs from different sources, they found that purines, the concentration, molar concentration of purines in DNA equals to that of pyrimidine. That was one wonderful finding. So, Watson thought that this Chargaff's rule, base pairing, this Chargaff's base composition would be clue in finding the structure of DNA. Therefore, these two scientists were analytical in their experiment. They analyzed data of crystallography, Avery-Chargaff's base composition, and they studied different they listened to scientists at different fora, and ultimately they came to a point to discover DNA as a double helix. Now, I want to say that Linus Pauling's contribution in this. Linus Pauling was a great molecular biologist, structural biologist. No one was comparable to him during 1950, but he suggested DNA might be a triple helix, but he could not prove. But the helical proposition of DNA helix, that also reminded Watson and Crick that DNA would be helical, and that was supported by the data of your Rosalind Franklin. But Rosalind Franklin, unfortunately, she died at the age of 37 during 1958. After analyzing data from different sources, Watson and Crick proposed the DNA as a double helix. They devised a model. That model is shown here. You can see both Watson and Crick. It is a landmark discovery in the history of science, as they proposed the DNA double helix structure or DNA model that made the road clear for different aspects of molecular genetics. As these two scientists proposed this model of a DNA double helix, this result was published in 1953 in the journal Nature, and for their great discovery, these two scientists along with Maurice Wilkins were given a Nobel Prize in Physiology or Medicine in 1962. But then this DNA is a double helical structure and it is right handed. When left handed and question of right handed comes, I think this diagram can help you to understand what right handedness is or left handedness is. When a helix is viewed from the top, if the helix run away from your eye in a clockwise direction, that is right handed. If it runs in a left handed manner from your eye, that is if it runs anti-clockwise, it is left handed. So, that speaks the left handedness and right handedness. Just to understand, DNA is a right handed double helix. Okay. It consists of two strands or two chains. The chains are called strands. Here you see the two strands are wound around an imaginary axis. Very often we think that the two strands are wound around each other, but it is not like that. Some people think that they are wound around each other just like a flexible wire, but it is not. But it is wound around an imaginary axis in a right handed manner. So, that is why it is a right handed double helix. The two chains are antiparallel. I will explain what antiparallel nature through another diagram subsequently, but this antiparallel nature of the double helix is determined because of the orientation of sugars. The deoxyribose sugar in one strand, the orientation is in upward direction, in the other strand it is in downward direction. That would be clear in the subsequent diagram, and therefore, when we write the two strands of DNA, we write 5 dash to 3 dash end, the other one in reverse 3 dash to 5 dash, because they are antiparallel. The bases of two strands are complementary. This sprang from that of Chargaff, Avery-Chargaff. His molar concentration finding purines equals pyrimidines, adenine pairs with thymine and guanine with cytosine. Purine always pairs with a pyrimidine. Purine is a double ring structure, pyrimidine is a single ring structure. Therefore, always 2 pairs with 1, making the paired structure 3. Never a 2 pairs with 2. Purine never pairs with purine, pyrimidine never pairs with pyrimidine. It is purine pairs with pyrimidine and same. Therefore, the uniformity of the DNA is maintained because everywhere purine pairs with pyrimidine. Then the 2 strands are held together by means of hydrogen bonds. This is key to many aspects. Adenine hydrogen bonds with thymine by means of 2 hydrogen bonds. Guanine with that of guanine pairs with cytosine by means of 3. So, AT has 2 hydrogen bonds, GC has 3 hydrogen bonds. Individually these hydrogen bonds are weak definitely, but if you look at DNA which is long strand, long chain having many base pairs arranged one above the other. So, many hydrogen bonds in the DNA make the DNA double helix very stable. Okay. Now, another feature, when we look at the DNA, its 3-dimensional structure, in the outer region lies the sugar phosphate backbone. So, sugar phosphate backbone are located outside the helix and the bases are in the core. So, the bases project inward from the sugar phosphate backbone. So, the bases form the core of the helix. Now, look at this diagram. This clearly indicates the 2 strands orientation of sugars are different. The O oxygen in the sugar at the top, you see in one strand it is upward, in the other strand it is downward. So, antiparallel nature of DNA is due to the puckering of sugars. This kind of arrangement is called puckering of sugars. And now, further the 2 strands are antiparallel, easily visible here. Next, the bases are arranged perpendicularly to the long axis of the helix. This is important. The sugar phosphate backbone is in outside and the bases they are attached to the sugar phosphate backbone, particularly the sugar, in a right angle manner. So, that is why they project in a right angle manner from the sugar phosphate backbone, and the base pairs stacked above one above the other form the hydrophobic core, and that adds to the stability of DNA. DNA is very stable because water molecules are excluded from the hydrophobic core. And further in DNA, purines and pyrimidines, its molar concentration is well explained, and pairing of purine and pyrimidine gives DNA is a symmetrical structure and its uniform diameter of 20 angstrom. And DNA is a helix. One turn of the helix has a pitch distance of 3.4 nanometer, and in a pitch there are 10 base pairs accommodated in a pitch. Therefore, the linear distance between 2 consecutive base pairs is 3.4 angstrom or 0.34 nanometer. And when we say the angular distance between one base pair and other, you might have seen a staircase. In a staircase, this is the first staircase, the second one is having a linear or a height, but it is having a rotation. Third one is like this. Similarly, one base pair here and the second base pair is 3.4 angstrom apart in linear direction, but angular rotation is 36 degree. So, this gives an idea how the helix is organized. In one side of the helix, if you look at, there are major and minor grooves. From one side you will find major groove, deep groove, and it alternates with minor groove. The grooves are important for accommodating or attaching with proteins. And when we speak DNA as nucleic acid, does it possess any carboxylic group? No. Then what contributes towards acidity of DNA or RNA? It is phosphoric acid. Phosphoric acid at neutral pH or just above that, around 7.4 or that, it exists in phosphate. So, out of 3 OH groups of phosphoric acid, 2 are utilized in formation of diester bond, but 1 OH group is free, that get ionized, and that adds to the acidity of DNA, and DNA having many negative charges is called polyanionic acid. Polyanionic acid, and these negative charges are essential for electrostatic association with positively charged basic histone proteins, subsequently we will be coming across. And sugar phosphate backbone is a constant feature. You take DNA from any source, whether it is bacteria or a virus or a plant, sugar phosphate backbone is common. So, that is why jemiti ame khata nauchandi, we are not worried when we are writing, we are not worried about the paper what it is, but we are what we are writing. Similarly, sugar phosphate backbone we are not worried, what we are worried about or serious about, what are the base composition, what is the base sequence. So, therefore, sugar phosphate backbone is a constant feature, but the variable feature among DNAs is its variability of a DNA sequence. The double helix of DNA described by Watson and Crick is a classical B DNA. Often it is written B DNA. What does it mean? B means biologically active form of DNA, and we know in our cells DNA along with proteins remain there in the nucleus, and water is there, salts are there. So, at a normal water concentration and salt concentration, the DNA exists in B form, biologically active form of DNA. That is why it is called B DNA, but the environment of DNA varies, salt concentration varies, proteins also in association in certain cases it varies. As environment fluctuates, therefore, the B DNA configuration also varies in certain ways. So, we will see that what the two important deviations from B DNA, but as the two scientists Watson and Crick, they worked together in developing the double helix structure of DNA. If one strand is called the Watson strand, the other complementary strand is called Crick strand. And what are the functions of a DNA? DNA is the molecule of heredity. We say DNA is the blueprint of life. Everything what would be there in the cell or in the organism, what kind of protein would be formed, when it will be formed, everything is written or imprinted there in the DNA. So, DNA in its base sequence stores the genetic information as the software of a computer does. So, DNA is often called the software of a cell. Then replication must DNA structure must justify how replication occurs, and we studied DNA consists of 2 complementary strands. When the 2 strands separate, each strand acts as a template by which another new strand would be synthesized. So, DNA explained this structure of DNA explained how from DNA another DNA or from DNA how RNA can be formed. Okay. Further, expression of genetic information, DNA not only stores the information, storing information is meaningless. The information, the genetic information must be expressed. That expression is in the form of proteins. Almost all genes express themselves in the form of proteins, as a result of which these proteins work as enzymes and carry out metabolic activities or cellular activities. And to the end, I want to tell you just three kinds of DNA are there in terms of structure. The DNA we study, it is BA-DNA, which is of intermediate shape, but A-DNA which exists at higher salt concentration, it is broadest and it is right handed. So, A-DNA, B-DNA both are right handed, but Z-DNA is left handed. We need not remember all the points of this, but for reference I have given here, but Z-DNA is left handed, B-DNA is right handed, A-DNA is right handed, and the repeating units of Z-DNA are dinucleotides, but repeating unit of B-DNA are mononucleotides. So, I hope all of you have gained some concepts regarding the discovery and the structure of your DNA, and this would further our understanding for DNA replication and gene expression. Thank you all.